Firstly, good evening, everybody. Thank you so much for joining. We have an interesting guest for the Technion UK webinar tonight. Before I introduce our special guest, I'm going to talk a little bit about the Technion and a little bit about what the Technion UK does. So you'll forgive me if I go back in a little bit of history and talk about Technion being established in 1912. This pretty much laid down the cornerstone for the Israel Institute of Technology and what would be known today as the Technion. It was the first institution of higher education in then Palestine, showing an understanding that certainly as Jews, the success of the Jewish state to be would heavily depend upon our ability to innovate through technology. The country's only significant natural resource is the brainpower of its people. Fast forward 100 years later and see how astonishingly that vision has been realized. Today, Israel is one of the technology powerhouses of the world, the startup nation, and where do the majority of these talented people and great ideas start? Yes, you got it, it's the Technion. Technion UK was established slightly later. It is still currently the leading British organization promoting, educating science, technology coming out of Israel, and fully supporting the Technion. Technion plays a vital role in every aspect of Israel's life. As you know, 80% of Israel's engineers are trained at the Technion. The Technion UK is a registered charity to support the Technion. It's one of a number of societies around the world whose function is to raise funds and promote the work the Technion is doing, its awareness, and all of its activities. Not many organizations can claim distinction on having been born in the House of Lords. Yet this is exactly where the Technion UK started, at a meeting in the House of Lords in 1951. Over the years, Technion UK has contributed to the infrastructure of the Technion, providing funds and support for buildings, auditoriums, research centers, equipment for faculties, for students and staff alike, sports facilities, student hostel, staff housing, social projects, on and on and on, specifically to support the work that the Technion is doing on behalf of the State of Israel. On the academic side, the Technion UK has endowed chairs and lectures, set up endowment funds, and established scholarships and fellowship funds in order to enhance the work that they do. So now that I've talked a little bit about the work that we do in the team in the UK, and certainly at the Technion, I'd like to address and introduce our special guest for this evening. Dr. Dedi Meiri is a worldwide recognized professor at the Faculty of Biology at the Technion in Haifa. Dr. Meiri studied as a plant biologist originally. He holds a master's degree in biochemistry, and he conducted molecular and genetic plant biology for his PhD, which I'm sure he can speak about a lot better than I can. During his postdoctoral studies, Dedi expanded his knowledge in human and cancer biology. Presently, his lab investigates the therapeutic potential of phytocannabinoids. His laboratory at the Technion is the only one in the world that has the ability to actively analyze active compounds in the cannabis plant. Please welcome our special guest. And should you have any questions, please write them down. And let's hear him talk about his work on immunity and his COVID-19 research. So without further hesitation, I'd like to present to you our special guest, Dr. Dedi Meiri.
Hi, thank you very much. I hope everybody can see me, and I will try to share the presentation. Is it already shared? Paul, can you tell me if you see the presentation?
It was on a minute ago. Let's see if we can get it on again.
Okay. So let's do this technical thing, and then we can start. So first of all, thank you very much, everybody, for joining us. Since I gave 2 talks in London to UK friends in the past year before this crisis started, I won't start to introduce myself too much or introduce the work. I want to go further and show things. For people that are joining us all over the world, from people that are stay awake all night to see me from New Zealand, and the people from the spaceships that are watching us. I'm sorry if I'm skipping things that the people from UK already heard, but I'm sure that I can answer there in the question that you will ask, and I will be happy to answer any question that will come. So doing that, we will go to we'll try to go to our presentation and start the talk. I hope you see the presentation. If not, Sophie or Paul will write me, I guess, or Sophie can mark it to me. Sophie, can they see the presentation? Is it looks fine? Okay.
Yes.
I assume that it's okay, and I will start. So I want to speak something like half an hour about the fact that cannabis is actually is part of a changing of a concept in the mind of medicine. Until few years ago, and for many years, medicine looked as one molecule, one target. There was the focus on finding a molecule, understand its target, and try to isolate it in the best way. Most of the medicines are one molecule that target a specific protein or a specific target in our body. And this is the way the FDA is working, and this is the way most of medicine is working. When Rafi Meshulam started to work with this plant, the cannabis, in the 60s, around 1963, 1964, he actually came back from Rockefeller, from his postdoc, and he works on terpenes. It's another molecule that are in the cannabis plant. And he thought that he can find this specific terpenes that call the specific feeling of the cannabis, the feeling of high or to be stoned. And he looked for that, and he actually found a different compound that he named them cannabinoids. And he discovered in the beginning the CBD, which he showed that is not a hallucinogenic and not causing the effect. And then a year after, he succeeded to find another compound similar to the CBD, and he named it THC. And he showed on monkeys and other animals that the THC is the right compound that caused this effect of the cannabis. And then there was a race to find what is the target. So we said one compound, one target. We find the compound that's causing the effects that we look for. Now there was a race to find what is the target. And just in the 90s, they discovered that actually in our body, there is endocannabinoids system. Our body produces similar compounds, and these compounds are called endocannabinoids. And the molecules from the cannabis, the THC, is just binding a receptors on our cells that actually our cells have that as part of our body and our system. And I won't go deep into the endocannabinoid system or the cannabis. Again, I guess most of the people that log into this talk are kind of knowing this kind of stuff, and I want to go further. So the endocannabinoid system, it's a system that balancing our body, the homeostasis in our body. It's a very complicated system. We know today to define more than 100 endocannabinoids that our body produces. There are few receptors. I will assume around 30 or 40 receptors for that. So it's a complicated system. And actually, in the year 1998, Rafi Meshulam and his team, that day his PhD student was Ben Shabbat, today a professor in Be'er Sheva in Israel. And Ben Shabbat and Meshulam published a paper called the entourage effect. And the entourage effect, they are actually saying that what they discovered that the endocannabinoids, there are two endocannabinoids, that every one of them is not very effective. but they work as in a synergism to activate the CB1 receptors. It's actually the entourage effect. The first paper in 1998 is not discussing the cannabis. It's actually discussing the endocannabinoids. But from there, they assume that the cannabis is working in a similar way. There are many compounds in the plant that actually working in a synergism to activate our system. So it's not one molecule, one target. It's few molecules, few targets, or few molecule, one target. But it's not a one molecule. So if we're coming back to the plant, we understand that the plant, then we know and understand that the plant not producing just CBD and THC, it produces many phytocannabinoids. Altogether, we can recognize today 10 families of phytocannabinoids, and altogether around 140 phytocannabinoids that we can analyze. And the same with the endocannabinoid system. So in order to start to work in my lab, we first develop analytical tools to analyze these molecules. Before that, we were kind of blind. We knew that there are a lot of compounds in the plant, but we couldn't analyze them, couldn't define them. So we developed for a few years analytical tools to analyze all the phytocannabinoids. And then we went further and we developed methods to analyze all the terpenoids in the plant. And then we go further when we develop method to analyze all the endocannabinoids in our body. And when we finished that, we were ready to start really to do research and to understand. And the idea of that, we can use equipment that doing high throughput screening. And the idea, we have robotics and few equipments that you can grow many types of cells in them and to screen many compounds and many different types of cannabis, if we're talking on cannabis, in very fast time. So this equipment, for an example from our lab, can take more than 1,000 images an hour, live imaging, and in 24 hours, you will get 24,000 images, which will go to the computer and you can analyze them and you can do them fast. So I want to give one example of what we're doing in the lab and then to take it wider. So for example, we were looking on leukemia cells, and in cancer is a given name to hundreds of different diseases, and we divide them by a type of the cancer, but also we can divide them by the type of the mutation that's causing these cells to be cancerous cells. So in this case, we took a leukemia cells which have a specific mutation that occurs in 60% of all the leukemia cancer. And we started to screen many types of cannabis. In my lab today, we have more than 900 different chemovars of cannabis. In this image that you see in front of you, every color, it's a different strain, it's a different chemovar of cannabis. And I would go deep in what we see. I will just say that if the line is lower, the meaning that more cells are dying. So you can see we screen many types of cannabis until we define one of them that killing these cells very efficient. We went further and we looked and we saw that this specific strain is not killing leukemia cells that don't have this specific mutation, and is killing the cell just when they are harboring this specific mutation. We went further and we learned about the cell biology, what is happening exactly inside, why it's blocking in the Golgi apparatus not to send this molecule to the membrane. I won't go deeper in the biochemistry and the cell biology, but when we understood the mechanism, how it's work, we went far. And we saw that if we're taking the mice and inject them the cancer and then the cannabis. So in this experiment, we inject the cancer and we waited 14 days that the tumors will start and then started to inject the cannabis. And we see that we save these mice and you can see that the tumor shrink dramatically. If we're doing a similar experiment, but now inject the cancer and starting to inject the cannabis from the beginning, the tumor is not developing as the normal tumors. because we're talking on leukemia cells, we create kind of leukemia in the mice while when we are radiating the bone marrow and injecting the cells to the bone marrow, and we got similar results that we saved these mice from this leukemia. Now we're asking the question, is it one molecule, one target, or is it, as Rafi Meshulam discovered in 1998, it's entourage effect, it's few molecules, or we need whole the plant, the whole plant. So what we are doing, we're taking the oil, and here you see an HPLC, high pressure liquid chromatography of the oil of this strain, and you see there are small peaks, all of them are cannabinoids. And what we're doing, we separate them, we fractionate them to few pieces. Here we fractionate it to 4 pieces, and we take every piece that contains some of these cannabinoids. So if we have 100 cannabinoids, now we divide to 25, 25, 25, 25, and we're going back to the cells with this fraction. And in this case, we saw that just fraction 2 give the same activity as the whole extract, and you can see it also here in colors. The red one is the whole extract, the yellow one is the fraction 2, and this is the other fraction that's not working. So we already eliminate 75% of the compounds, and we're going further. We're taking this fraction here that works well, and we defractionate it, we refractionate it again, and we did it again and again until we discovered 3 molecules. You see them here, C1, C2, C5, 3 cannabinoids, that if you put them on the cells or in them or gave them to the mice separately, purified, you get exactly the same action. Any combination of each one of them or combined together, not giving the same activity as the whole 3 of them. And if we take the 3 of them purified and inject them to the mice, we're getting the same results as we got with the whole plant. You see, it reduced this tumor dramatically. Moreover, we succeed to take these tumors and to extract from them the phytocannabinoids, and we discovered that these 3 phytocannabinoids, C1, C2, and C5, is entering to the tumors and accumulating in the tumor. So even though these mice have these tumors on their back or as leukemia in their blood, and they're getting the cannabis to their intestine, still the cannabis is reaching the tumors and accumulating there. So what I showed you very fast now is actually that we can take the plant, we can screen type of disease. In this case, it was cancer, define which plant is working the best, and then going deeper and deeper until we find which compounds are working the best. I can show you this on different diseases. What we're showing here, it's how we're creating multiple sclerosis in the mice, a different disease named multiple sclerosis. And now we're treating these mice and see how the cannabis can help the symptoms of multiple sclerosis. And what you see here is the control. The mice are developing the disease around after 15 days of treatment, and you see the disease become very severe, and the line is straightening. And then we treat with cannabis, and we see that there are few strains of cannabis that improve the disease, but there are different strains, even though they're all kind of similar, have the same amount of CBD and THC, they improve differently the disease, all of them improve. But we also have strains with the same amount of CBD and THC that make the disease worse. So what we need now is to going deeper and deeper to fraction the oils of the blue one or the yellow one, and understand why in the blue line, the strain from the blue line, make it better. What are the compounds on top of the CBD that helping this in this strain to improve the disease, and what are the compounds that make it worse in the yellow one? So we can go on this research and do that to any diseases that we actually wanted to research. And in my lab, I have a model, cell models and mice models for a sleep disorder, for multiple sclerosis, for Alzheimer, for epilepsy, for cancer. We can do that, the same idea, for many different diseases. But more than that, we can do that not just for cannabis. I have one student in my lab, a PhD student. They collect more than 90 different algae from the Israeli shore, extract from them the active compounds, check on different types of cancer, breast cancer and lung cancers, how this affects, define one algae that was very effective killing these cells, and now we are trying to define which compound on this algae is working and doing this effect. We can do that with mushroom, we can do this with many different plants on many different diseases. In order to do it more efficient, we actually opened together with the Technion a company. We named it MyPlant. That in this company we create many models of diseases. We want to create 20, 30 models of diseases, which we can do high throughput screening. We can screen very fast, and we are giving we're giving a service. We're saying to the cannabis growers or to other people that working with different plants or different materials, send us your materials. We will screen them on all these diseases, and we will give you a report. We will tell you if your plant or if your extract, if your materials that you took from the algae is the one that working the best, or if it's one of these ones. But doing that on many diseases with many compounds, we create a big database, and then we can do what we call AI, artificial intelligence. We can start to look on big numbers on many diseases with many compounds and to look on that. So going to the coronavirus, it's actually the same. We have a virus that binding our lung cells and getting into these lungs by binding a receptors, and we understand the cell biology and what exactly happened there. So what we did, we said, okay, let's build a screen for the virus. Let's build a screen that defines on lung cells if the virus can bind. In order to bind, the binding a specific receptor called ACE2. So we can look on these lung cells if treatment with cannabis or different plants reduce these receptors, and now it will be more difficult to the virus to bind. Going to all our data that were already collected in 4 years, we went and we started to look in which plant, which cannabis already reduce these receptors, and we find we have a preliminary result showing that there is specific strains that can reduce these receptors. We can do that on the cytokine storm. Actually, when the virus is entering our lung, our immune system recognizes it and send the soldiers to attack it. Sometimes the attack is so severe that it's harm to our body. It's called a cytokine storm. Actually, the people that died from the virus, from the coronavirus, the COVID-19, are not dying from the virus. They are dying from the immune system, our own immune system that attack it so severe that it's caused us for death. So we can do the same thing. We can take these cells of our immune cells, the macrophage, the T cells, and we can define which types of cannabis or which types of plants or materials reducing this specific compound that call the immune system to attack and to reduce it. So what we did, we create 4 different cell models for different approaches of the virus, of the coronavirus. One of them is how to reduce the ACE2 interaction with the virus. One of them, how to reduce the virus protease activity, never mind the details. One of them, how to reduce the ACE2 expression of the lung cells. And one of them, how to minimal the effect of the inflammatory response of the immune system. So actually, having the ability to do high throughput screening and have all these cell models on one hand, from the other hand, having the ability to look on a pool of a variety of compounds together, like I have in the cannabis or in the algae, and then to reduce it and to define which compound doing this effect, give us the ability to actually to ask it in almost every disease that we can create a cell model. And to come back to what I started to say in the beginning, the cannabis is open our mind that actually we can start from a plant that have a lot of molecules and started to reduce them and find the minimum compounds that doing maximum effect. It doesn't mean that it needs to be 1 compound, 2 compounds, 3 compounds, we don't know. It might be that in many plants, like in mushrooms or like in algae, similar to the cannabis, we will get an entourage effect. We will get an effect that is more complex than 1 compound. And it started before it's actually started a little bit before the cannabis. We know that today in cancer treatment, in many treatments, we combine many types of chemotherapy and treatments together and doing a cocktail. And it's actually started even before when treating the HIV virus, today treating with the cocktails. And more the medicine started to be a combination of compounds together that actually activate different pathway and getting synergism. The cannabis is actually giving us, it's opening for us the door to ask that not just for that plant, but for other plants that are now arising. And if people are knowing, I'm not mentioning mushrooms without a reason, there is now a movement about a few mushrooms that are starting to emerge as, again, a medicine that have many compounds that doing a synergism. So before we're going to questions, I just want to say a big thank you to the team of my lab that doing this amazing work of this research on cancer, but also on epilepsy, Alzheimer's, sleep disorders, and many other diseases. I want also to mention Cannasol. It's a company that I have together with the Technion in Caesarea and my plant, that part of the things that I showed, they help on that and did. And Sophie, I think I'm done and we can go to question. Thank you very much for everybody that listening. It's feel like crazy to speak to yourself and in the computer, but I hope there is somebody out there that's listening.
Yes, hello. Everybody can ask their questions. There's a Q&A in the bottom. Please type it for me and I ask Dedi. So Dedi, the first question for you is coming from Rose Klein. What is the current state of the art in testing patients' DNA and predicting how a person may respond to cannabis-based treatment, and how is personalized the treatment? How to personalize the treatment?
Okay, so we need to separate it in 2 things. In the DNA is a cooking book of how to build proteins. First of all, endocannabinoids are not proteins, just the receptors are proteins. What we can define from our DNA, if there is a mutation, if we have a problem, and Ethan Russo in the 90s published few papers about endocannabinoid deficiency. It's when we have a mutation in one of our receptors, and for that we can know it can cause a disease that maybe we can cure. But we can't learn from the DNA of a person how these receptors are expressing in different cells, how the endocannabinoids is the diversity of the endocannabinoids, because they are changing during the day. In order to do personal medicine, we need to analyze exactly the endocannabinoid receptors, and this we can do just from a biopsy and not from the DNA.
Thank you. Roland G is asking, how close are you to developing and testing a vaccine for COVID-19?
Very far. I got approval from the Technion to open my lab 3 days ago, and we're starting to work.
The molecule is specifically found in cannabis, or it can be found in other plants?
Not that I'm familiar. Cannabinoids are produced mainly in cannabis. I'm familiar with another 8 different plants. 5 of them are from the Amazonas that can produce cannabinoids, the specific compounds, but they are producing them in very, 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 very low amounts. So 1,000 times less than in cannabis. The only plants that produce these compounds in high amount is cannabis, at least as far as I'm familiar, and I think I quite know about that. So it's producing just in the cannabis. Of course, we can try to produce it, and there are many companies that are doing that today to produce it in yeast, or produce it in small algae and other things, or to make it synthetic. But meanwhile, we can produce it just from cannabis.
Tim is asking, how do you proceed if the compounds that we needed as a combination are in different fractions? When do you do the 25% fractions in the beginning of your process?
So if I understand right, you're asking what is my methodology to narrow it down. So first of all, I'm going on the big compounds. So usually it will be THC and CBD, and separate them to see if they are not the one that doing the major effect. So I will cut it in a way that I will have the CBD or the THC alone, and then the other parts alone. And from them, I'm going from the results. I see where I'm getting the best results and going there.
Okay, so Nitzan Levinson is asking, hi Daddy, you talked in your presentation about fraction with different cannabinoid composition. You did same process with terpenoids?
Yes, so in order to not to make my talk too complicated, I'm talking about just cannabinoid. But in the same fraction, I have cannabinoids, terpenoids, and flavonoids. And we look in all of them. So I look on them, I don't know which compounds making the effect. So in a specific fraction, I will have 14 cannabinoids, 8 terpenoids, and 3 flavonoids, but we're looking on all of them. I'm not deciding. So the answer is in general, yes. We're doing the same thing from terpenoids and flavonoids, but we're doing it together. I don't need one fraction for that and one fraction for that. In the same fraction, I have all the active compounds.
Boaz Wachtel is asking, did you check the antiviral effects on ibogaine on COVID-19?
So tell Boaz not yet. And Boaz, it's a big honor for me that you are listening to my talk. Thank you very much. If somebody don't know Boaz Wachtel, Boaz is the face of cannabis in Israel. We have Rafi Meshulam, which is the professor, and Boaz Wachtel, who was the advocate. He was the head of the Green Party, and we own him kind of the medical cannabis in Israel.
Nice. David is asking, what are the C2, C1, and C5 compounds?
2 of them are known compounds, and one of them is unknown. It doesn't even have a name. We are naming it now. We are publishing a paper now with all these 3 and name them. So 2 of them are known compounds, and one of them is unknown. I learned that it's better not to say the right names, because then cancer patients starting to look for that and I think it's irresponsible for me to say the compound. Then cancer, it's a given name to hundreds of different diseases, and each one of them have different panel of mutation, different panel, and the treatment should be very, very specific. And what's happened when I'm saying that, people starting to stop using their medicine, and I think it's irresponsible for me to say that. It's not that I'm hiding something.
Patricia Zimet is asking, are you planning to study curcuminoids?
What is
it? Curcuminoids, yeah.
I didn't think about it yet, but why not?
Okay.
It's all a matter of funding in the end.
So Philippe Taron is asking, does medical cannabis actually cures or only reduces the disease it's designed for?
For the cancer, if it's cure the cancer or just...
Or just reducing?
So in the mice, we never check to the end because there is a limit of how much in the control the tumor can grow.
Mm-hmm.
And there is a limit how much we can check. We are raising now money and taking it to clinical trial. I guess in few months we will be in clinical trials in humans, and we will have better answers for that. In the mice, we never reached to the point that we totally curing it, but it's because of the control that the tumors become too big.
Okay. Thank you. Juan Pablo is asking, why some cannabinoids are not totally bioavailable? Is it true that CBD is absorbed only 30%?
I didn't understand the question again.
Okay. Well, how come cannabinoids are not totally bioavailable?
Okay.
Is it true that, I'm sorry, the rest 30%?
Yeah. So cannabinoids are lipophilic, they are oily. And our body is mostly built with water. Our blood and everything, we are 80% water, not 80, but similar, 80% water. And for that reason, the bioavailability of cannabinoids is quite low in our body. And in order to improve them, we need them to be more dissolvable in water. And there are many companies and people that are trying to do that. But this is the reason that the bioavailability of this compound is very low.
Kevin Evans is asking, hello, Dr. Meiri. I was wondering if you are using AI techniques such as a quantitative structure-activity relationship modeling software, and if it's helping to narrow down the possibilities.
Yeah, so part of what my lab did in the last 4 years when we started with cannabis, we did a database project when every cannabis that's getting out to a patient in Israel in the last 4 years is coming to our lab first, and we're doing full analysis to this cannabis, the cannabinoids and the terpenoids. And on the other hand, we are follow up on patients. We are follow up on 5,000 patients, and we're trying to do so we're talking about 90 different strains with a lot of compounds inside and many indication, many patients. So on that, we're trying to do what this person, I sorry that I don't remember the name, just asked. We're trying to do AI in artificial intelligence and to narrow down and to get good ideas where to start. So let's say if we want to start a research on sleep disorders, we first look on which strains and which compound worked the best on people that already took that from this analysis, and then we are going mice model to check it. And the same thing, we are using AI, artificial intelligence, in order to learn how to do the screening faster and to define all the molecules, these cannabinoid, terpenoids, and the endocannabinoids in our samples. So we're using that, and we have people that are doing that.
Josh Frovine is asking, is it more effective for the cannabinoids to protect the immune system from the cytokine storm, or to target the coronavirus itself? Can the cannabinoids target the coronavirus itself?
So I have no idea about directly antiviral effects of cannabis. There are a few publications about that, but none of them show really strong antiviral. So I don't think that the cannabinoids will attack the virus directly. And also, I don't think cannabis can be, the medicine that's curing the world from the coronavirus, or replace, immunity or something like that. What cannabis is known to do, he's known to balance down the immune system. So if you are infected with corona and you will use cannabis, it might be, I wouldn't suggest to anyone to try that before we finish the research, or people will prove that, but it might be that it reduce the activity of the cytokines, and it won't, our body won't develop the cytokine storm, and actually you won't cross from a mild patient that just feel a little bit sick to a severe patient. On the other hand, and we have a preliminary result for that, it might be that the cannabis reducing the expression of the protein that the virus will need to recognize on the cells. So it will reduce the infection of the virus to the cells if what we think is true. Again, it's so in the beginning, just an idea and just a beginning of a research and not something established.
Rosaria Ricci is asking, how can you persuade clinical researchers that are not familiar with cannabis research to start clinical studies in humans with combination of different cannabinoids targeting specific tumors? And is it the Technion willing to provide data of clinical studies abroad and function as a multinational preclinical research center?
So the Technion is a university, and the technology in university is not clinical hospital, but it's aligned with Rambam Hospital. Actually, our medicine faculty is sitting in Rambam Hospital. And together with the hospital, we can do a clinical trials. In Israel, we have 60,000 patients that getting cannabis under prescription. And I'm alone participate today in 11 clinical trials regarding cannabis. Just for example, we have a clinical trial in Afula Hospital that how cannabis can help in neuropathy in cancer patients. So we are doing that already, and it's doable, and at least the medical or the physician in Israel already treating patients from 2007, and in Israel, it's not a problem. If I'm coming with a good preclinical data like I have, it's not a problem at all to go to clinical trials.
Chris Matthews is asking, have you seen a high degree of variance in the therapeutic potency of individual compounds between strains? What is the most surprising result you have seen in the past few years?
So if I understand right, he's asking if the same compounds from different strains can act differently. And the answer is yes, but it's more complicated. So if it's the same compound, it's the same compound. The question, if our analysis is not wrong to think it's the same compound. So I will give an example. If you're taking two strains, both of them have high CBD, but the CBDs act differently. When we are doing a deeper research, we realize that this CBD is different. It's actually isomers of the same compound. So they are not really the same thing. And for that reason, they can act differently. Again, this is a research that is just in the beginning. We're just discovering this kind of thing. So I don't want to say it in too confidence, but the answer is yes. We can see compounds that we thought they are the same, but when we are going to NMR and we're going to a deeper and better equipment checking that, we realize that not really the same compound.
Guy is asking, do you fragment monoterps and sesquiterps? And do you search for specific molecules that target the autoimmune disease? Also, do you study the plant's roots as well?
So for the first two questions, the answer is yes. We separate also monoterpenes and sesquiterpenes, and we are checking specific molecules that we suspect that they are causing this effect. So for both questions, the answer is yes. Regarding the roots, I did few works in the past regarding roots, and it was with a Better Company, one of the growers in Israel, that I got roots from them, and we tried to examine them. We didn't find any cannabinoids in them, and we didn't see on the cells that we checked any major effects. It doesn't mean that we checked the right thing. It doesn't mean that we did it properly. It doesn't mean that the extraction was good, but this was my result.
Dedi, I see a lot of questions about the cytokine storm.
Okay.
Natalie Kat is asking, can it help once the cytokine storm has already developed? And there's another question from Calypso Lara. There are a lot of similarities of effects with cocaine on the cytokine storm. Only the adverse effects are absent. Can you talk about it a little bit?
Yes, so I will go back to that and try to explain the idea and why it's look maybe promising. One of the known effects of cannabis is balancing down the immune system. There is a lot of work regarding that, and also my lab doing a lot of work of how cannabis affect the immune system. The reason that cannabis is working quite well to autoimmune disease like gout or like lupus, or it's working quite good in IBD, intestinal bowel disease like colitis or Crohn. The reason is because it's balancing down the immune system. When the immune system is highly active, the cannabis is balancing down. So we have drugs that can shut down our immune system totally. And when we have autoimmune disease, or our body is acting too strong, in the hospital, we can give these medicines that shutting down the immune system totally. The problem with that it's not so good to shut down our immune system totally, because then we are very, we are opening all our defenses. So one of the first and pioneer work with cannabis showing this effect was with the bone marrow implantation, which they showed that in bone marrow implantation, if you are giving high CBD or CBD, the rejection of the bone marrow is much lower, and for that reason, and the reason is because our immune system is less vulnerable, less attacking the new bone marrow than being implanted. So that become a good treatment, because you're not shutting down the immune system, you're just balancing it down, and it's reduced the rejection. If we're going back to the virus is actually causing something similar to autoimmune disease. The virus is entering our body, attacking the cells. Our immune system recognizes that and running there, but it's running like crazy. Sometimes it's so active that it's starting to hurt itself. It's causing a pneumonia. It's causing the disease itself. Like, people are not dying from the virus, they're dying because they can't breathe, because of the infection and the action of the immune system. The immune system, the cytokines are molecules, they are messengers. The cells, the immune system reaching the place, recognize the virus, recognize the attack cell, and you need to call his other troops, you need to call the other soldier. So he sent the messenger, he sent the pigeon with a message. This pigeon, this message, it's a cytokine. It's a small protein called cytokine that going in a diffuse and actually calling the other cells, the other T cells, the other NA killers, the other immune cells to come to this area. If we were balancing it down, if we would reduce the tone, he wouldn't call 2,000 of them, he will call just 200 of them, we will solve the problem. We know that cannabis can tune down the release of the cytokines. On that thinking, we are assuming that cannabis can be helpful in order to reduce the activity of our immune system in the virus attack, and for that reason, to save many lives, that there won't become a severe patients that have a problem to breathe of their own.
Thank you. Jennifer Stamps is asking, do you see a future for isolating therapeutic compounds from both cannabis and fungi and combining them for diseases that have multiple pharmacodynamic targets, or perhaps combining them for personalized treatments for patients with multiple issues? So cannabis and fungi.
Yes, so first of all, yes, I think in the future, and this is part of what I'm building in my plan, checking many diseases with many compounds, that with what we call artificial intelligence, when we will have enough data, we'll be able to start to combine compounds from different origins, one from fungi, one from algae, one from cannabis, and getting a synergism between them. Again, you know, if we're going hundreds of years ago, you know, the medicine, the people that cure people, we went to the forest, pick a leaf from here, a leaf from there, a leaf from here, and then smash them all together and gave you as a medicine. So usually they wouldn't use just one plant. If you're going to the Bushmen and you're going to the people that treating traditional treatment, or even the Chinese treatment, it's usually combining different plants together to getting this synergism. It's very hard to do this movement. I'm working in the Technion. We are the most accurate square place in Israel, and I need to do very accurate science on that. So I don't want to take a leaf from here, a leaf from here, a leaf from there, that is one times have this compound, one times have that compound, and changing all the time. I want to reach to understand this combination, but in very accurate way, to understand this compound is important, doing 30% of the action. This compound is important, doing 20% of the action. This compound doing the last 40. This is binding these receptors, entering the cell here. This is doing, to understand that, but then to know how to combine that and to take it to a real medicine. I don't care if in the end they will take a leaf, and a leaf, or make it synthetically, or making whatever. But we need to learn that and to know, and I think we have the right path for that.
Anuj Desai is asking, how important is the THC-CBD ratio in effectiveness? Can you have two strains with the same ratio that have very different effects?
Yeah, and I showed it in few results that I had today. I showed that in the multiple sclerosis disease, that I took five plants with the same ratio of THC and CBD, and every one of them act differently. I can say that in the leukemia, I can take the same amount of THC and CBD, and it won't affect because the compounds are not the THC and CBD. So it depends what are the compounds. The ratio between THC and CBD can be important just in illnesses or effect that the THC, the CBD are responsible for that. In many cases, it's not just for them, it's not them or not just them.
David is asking, Dr. Meiri mentioned it's the overreaction of the immune system that causes the problem with the coronavirus. In your studies of the disease, do you find the immune system is often the problem, not the disease itself?
First of all, I want to do a disclaimer. I'm far from being an expert for corona, and I'm afraid to make mistakes or to say wrong things, okay? I'm just starting the research in my lab on that, and I have almost zero results. I have preliminary results from our previous work on different things. So when we're doing a research on multiple sclerosis or Alzheimer or on epilepsy, I'm also checking the immune system. So I have data on that. When I'm doing proteomics or RNA-seq on lung cancer, I know ACE2 receptors and I know that it's reducing, but I don't have enough research or study to answer these questions, and I'm not an expert enough to say that this is the only problem that the virus is doing or things like that. So to answer the question, there are many diseases or many actions in our body that our immune system is overactive, hyperactive, and cytokine storm, it's not something that the coronavirus invented. It's an effect in our body that can happen in not just in our lungs, in our other places. It's a known a problem that viruses or bacteria or other infection can cause.
Trina Hazak is asking, based on the research you showed, some chemovars show pro-tumorigenic effect and other anti-tumor effect. Are there any certain tumors that you wouldn't use cannabis in, or certain cannabinoids or terpenes that you are more concerned causing pro-tumorigenic effect and we should avoid in the cancer patient?
I don't have a good answer for that. I think that there will be cancers that cannabis can more activate than blocking. And it depends on the mechanism of the cancer and which cannabis we are using. Cannabis can be a blocking proliferation, but we know that it can also cause proliferation. It can block migration of cells, but we know that it can also activate migration. It depends on the types of the cells and the types of the cannabis. So the answer is no, I still don't have a specific types of cancer that I would say I wouldn't use cannabis with it. But on the same path, and when I gave talk in London, I talked about that. In two studies that we did with Professor Gilbert Sella from Emek Hospital, the head of the oncology department, we saw that when cancer patient being treated with immunotherapy, so immunotherapy, if I'm flattening it a little bit, is when we are getting drugs that activate the immune system to attack our cancer. So when we are getting immunotherapy, we actually hyperactivation the immune system to attack the cancer cells. And as I told you before today many times, the cannabis is tuning down the immune system. So you're giving a therapy that activating the immune system, then you're giving cannabis that tuning it down, which blocking actually the action. And what we are showing in two studies that we did, one is already published and now one is published now, that it's still, I would not suggesting somebody that taking immunotherapy to take cannabis with them until we will define exactly which cannabis you can take and which one is not. For now, as a general, I would say that it's more risky than helpful to take cannabis with immunotherapy.
Okay, I have one more question. Jerry Cutt is asking, does the extraction method impact the effectiveness of the cannabinoids?
Yes, so the extraction method will take out or change the pattern of the phytocannabinoid, terpenoid, and flavonoid. If we're using butane extraction versus a CO2 extraction, ethanol extraction, press, and methanol, every one of them will actually extract different families of cannabinoids, terpenoids, and flavonoids, depending on the polarity of these compounds and other function. And for that reason, it can change the medicines that we're creating. So it can elevating or reduce specific compounds in a different specific extraction. So the answer is yes, the extraction method is important for the output or the outcome that we will get from this extraction. I don't know to say which extraction method is better or worse. I can say what is the difference between them, but I don't know. So if I know what is the compound that I need to extract, then we can find which extraction method is for the best for that. But if it's still a black box, I don't know which extraction method is better. I just know that they are different. And we did a lot of work about that. We're writing now a paper about the stability of the compounds and the shelf life. And on that, we did also different extraction methods. So we did a lot of work regarding that, but I don't know to say good and bad.
So Dedi, you answered more than 20 questions and I have 30 more, but I think
I will be happy, you know, to come again to answer questions if it's something that you want to collect.
I think there are a lot of questions out there. Sorry. Yeah. Why don't we get back to the people who were on this webinar today, and we might be able to offer another occasion for Dedi to answer the remaining 30 and more questions.
I will be more than happy to answer any question and to spend more time here. You can come back to the people on the webinar and suggest an hour, and I will be vacant there to answer the questions.
Thank you so much, Dedi. Thank you.
It was my pleasure. Again, thank you very much, all the people that listen and stay with us. And keep safe and healthy. And I hope to meet everybody that I know soon and get from this situation. Thank you, Sophie.
Dedi, I want to thank you.
Me. It's a big honor to be here. Thank you very much.
Dedi, thank you so much. You and the Technion do some phenomenal work, but let me give you a piece of science you haven't worked out yet. The longer you talk, the more questions are asked. You haven't worked that one out yet, have you? Okay. That just means it's purely success.
I want to say one thing which is important. In the Technion today, there are more than 30 laboratories that working regarding the coronavirus. There are amazing things being done. I saw yesterday a good friend of mine that she created a diagnostic kit, home diagnostic kit that you can, you know, spit in the tube and know that if you're sick or not in less than 1 hour. And actually, it's very simple and smart. You just need to think about it. And there is a better diagnostic. There is invention of equipment to the medical, you know, teams. There is amazing work being done in the Technion. I think I'm just starting now when there are few labs that are already working 30 days very hard. So Technion is doing a lot on that, and I'm very proud of what we are doing.
Dedi, I've said this before, we thank our health staff for saving lives, but we don't get an opportunity to thank you and the scientists and the staff and the people at the Technion working day and day night who are saving lives. And I hope one day you'll have somebody who comes up and says, thanks to your research, I'm alive today and my children are alive today. So we all are waiting in anticipation for you to figure out the solution. You clearly are an intriguing and motivational and inspiring person amongst your team. We are always in awe of what you do and how you do it. Please continue doing it. We will support you. We will stand behind you and the Technion, and anything you need to help you, help us, we're here. So kol hakavod, well done, stay safe, tiyeh b'shalom, and hope to speak to you soon. Thank you.
Thank you very much. Bye-bye.